Good morning, and thank you very much, Cameron, for this invitation. When I looked at the preparation of the programme, I see that there's quite a few people, both this morning with Rob Don and also this afternoon, will be talking about drug discovery programmes for Leishmaniasis and other trypanosomatic diseases. And when I started thinking about what to talk about, actually, the title is slightly wrong. Designing new treatments for Leishmaniasis is perhaps incorrect. It really should be thinking about new treatments for Leishmaniasis, because when I started to look back and review where we were and the progress we're making, then I sort of realized that there were quite a few gaps and also some lack of direction in which we should be traveling. So the first thing, for those of you who are not Leishmaniacs, is just to remind you that Leishmaniasis is a complex of diseases. There are 17 different species that cause this disease, and probably there are people in the audience who will tell me more than that. But in terms of clinical manifestations and pathology, it's a complex of diseases which ranges from visceral forms to cutaneous forms, and I'm going to talk about all of these. And clearly the demands for drugs that work on the skin and drugs that work in the viscera are very different. And also, we've shown over the years there's a lot of species variation in drug sensitivity, which is also an important thing. So I think the challenge is this is not one disease, and there will not ever be one treatment. We're going to have to come up with probably several new treatments for this disease. But again, for those of you that don't work in the area, just a little assembly of all the current treatments or drugs that are available. And it might look from this little list here that there are plenty of choices and plenty of opportunities. But I have to say most of these drugs are very toxic in their nature, have poor routes of administration, and also very variable activity against different species. So not is all well, but in the last 15 years, the liposomal formulation of amphotericin B, miltefosine, and promomycin have all been registered for use in the Indian subcontinent. So things have changed. There's been some progress. There's been some improvement. But when I was sort of sitting in the bar thinking about what to talk about, I thought that there are 5 areas in terms of visceral leishmaniasis, I'll come on to cutaneous later, that really have to be addressed pretty urgently. And I've sort of got one slide on each. There's the need for a safe oral treatment. There's the need for drugs which are also active in East Africa as well as India. There's the ever-increasing problem of HIV co-infections and what to do about them. We've shown that drug combinations can work, but we haven't optimized them. And also I'm not going to talk about improved formulations today, but there's opportunity for improved formulations. But I think really what we need to start with is just coming back to some of the issues and the priorities which I think we really need to focus on. So in India, Bangladesh and Nepal, there is an aim to eliminate this disease. That means to reduce it to, in terms of incidence, less than 1 in 10,000 of the population by next year. It's now been extended actually to 2017. But the drugs that you need for an elimination program are actually quite different from those that you need for necessarily patient treatment, because not only has the possibility of elimination program got to include safe drugs that might be used in people who don't actually show visceral leishmaniasis or Kala-Azar. But there's also got to be, if you're wanting to interrupt transmission, treatment of people who have post-Kala-Azar dermal leishmaniasis, and also to disinfect, that's my terminology, asymptomatics, who are actually the reservoir for the disease, as well as having this safe, sustainable post-elimination drug. Because in fact now we should be planning for post-elimination period. And actually, I don't think we have any drug in that little armamentarium that's suitable. The one drug that has been proposed is the liposomal form of amphotericin B, which I won't go into in too much detail today, but just to say that there are 3 key issues with this particular form of treatment. Though it can be used as a single course of treatment for the treatment of visceral leishmaniasis in India, there is a need for a cold chain, and those of you that have ever visited primary healthcare centres in Bihar state in India will know that that is an imaginary concept. There's a concept of cost, because it's still, even on cheap WHO rates, it's still $18 per 50 milligrams of an ampoule, which means you're still talking about $100 per patient, which is way out of anything. And then this was given to me by Rob Don, actually, a slide which comes from DNDi, which shows that though it is in the publication said this is a single dose treatment, actually it's a single complex course of treatment that has to be administered by trained people in a primary healthcare centre or another hospital that has a proper cold chain and proper facilities available. So though this is a very useful treatment, is this the right sort of approach actually for sustaining an elimination program? And I have to be a little bit honest and say no. The second key issue, which really comes in our thinking about treatments for visceral leishmaniasis, are that all these drugs that have been registered, promomycin, miltefosine, and the liposomal amphotericin B, that have been highly successful in Bangladesh, Nepal, and India, in East Africa, there's been significant failure of all these drugs. So we have to ask ourselves a very important question: what is going on in Sudan in particular? There's some gaps here. Actually, one of the things that really struck me, I spent hours preparing this, Cameron, so going back through all the literature. I cannot find a single paper where amphotericin B fungizone has ever been tried in East Africa. So there are still some gaps there in our thinking. But if we look at the percentage, this is at 6 months and after course of treatment finished, and compare with this particular columns, 39, 76, 60%, way below the acceptable 90% cure rate, we have a major problem. And we have to try and explain the differences. And we know that all the pathology and the things, there is a difference between Sudan and the Indian subcontinent in the way the disease develops. We also know from people, some in the audience, like Jean-Claude Dujardin, but this comes from Gabi Chernian, this picture, that actually the parasites in East Africa, Leishmania donovani parasites, can be distinguished by molecular taxonomy from those from India. But actually, you take those parasites and you shove them in a macrophage in vitro or a mouse in vivo, and they have exactly the same drug sensitivity profile that you find from the Indian isolates. So that's not the explanation. The explanation must come somewhere from pharmacokinetics and pathology of the disease and the patients in those two populations. And it's clearly an area which requires urgent research, because I think we're going to be rolling out new drugs, as Rob Rodon will probably be talking about new drugs, and we might face just the same situation, that drugs are working in one continent but not the other. We have to have an understanding of the drug, how the different patients, different populations handle drugs. And this is just from an ex-MSc student of mine, Ripu Bans, who did a PhD on pharmacogenomics, showing that, in fact, our understanding of how drugs are handled in Africa compared with Asia, where there's a big gap of information, is really pretty primitive. The third area where I think we're missing something is actually in the ever-increasing number, both in India, where it's about 5% of cases now are HIV co-infection, but in northwest Ethiopia is now 25% of patients are HIV visceral leishmaniasis co-infection. Is actually understanding how best to use antiretroviral treatments and anti-leishmanial drugs. There have been many studies done over the last 15 to 20 years on treatment of VL HIV co-infection cases, starting mainly in Spain. And really what we've been doing is just empirical studies. We try this anti-leishmanial, it knocks down the leishmania. We try this heart regime, and it knocks down the HIV. But there's actually no proper systematic study being done on drug interactions, both in relation to how they might work together, because we know some protease inhibitors actually kill leishmania, but also, most importantly, and I've taken this from the Liverpool University website, in terms of drug-drug interactions, where Liverpool University has in terms of antiretroviral drugs, has worked through drug-drug interactions where the side effects might occur between a known type of drug, like in this case it's the anti-malarial drugs, side effects might occur. So where there's been no systematic studies have been done in this area. And again, in terms of thinking about what should be done, if this is an increasing population, this becomes a priority. Another area of work that's been done is studies that actually Rob Don and I started 10 years ago in DNDi was to look at drug combinations. And in fact, drug combinations are using these combinations, ambisome and promomycin, ambisome and miltefosine, promomycin plus miltefosine, the details of which are irrelevant here. The principle is important. All these drugs were used in the clinical trial at the doses that are normally used as the drug alone. So high dosing, but for a shorter period of time. So it was a 7-day or 10-day treatment. So it was to shorten the course of treatment. But what's never really been done, and again is an open door for further studies, is to actually take this further and work out, again, looking at those combinations, at proper drug-drug interactions, in analysing sort of the PK/PD of the drugs alone and then in combination, to see if it can be even shorter course of treatment, but also reducing the dose, because we've still got two expensive drugs here, ambisome and miltefosine, and anything that could reduce the amount of drug to be used in the combination or to make that combination more effective is actually worth looking at, because it does, say, shorten the course of treatment significantly from the normal 28 to 30 days down to 7 days course of treatment with those combinations, and therefore more thinking about plasma tissue concentrations of drugs used in combination is clearly an open area for further treatment improvement. So I won't go on about formulations here, but obviously there are ways of improving formulations as well. But as I was trying to stick to my 12 minutes to keep Cameron happy, I will move on to the next disease. So I think there's still plenty of challenges there for visceral leishmaniasis, and we'll obviously hear about probably the newer compounds becoming drugs in the next few talks. But there's this other disease. So if visceral leishmaniasis is a main focus for many of the funding agencies and the drug development agencies like DNDi, who've clearly taken a strong lead in the area, then for cutaneous disease, which is actually at the moment pretty rampant in the Middle East, the disease that was under control in Syria 10 years ago is now out of control. Afghanistan with over 200,000 cases per year, and it's still heavily embedded in South America. But one of the most important things about cutaneous leishmaniasis is it doesn't kill people, it just disfigures and stigmatizes them. And there's been very little investment. But investment will only really come if there's a clear strategy, which we don't have. Well, obviously, personally, I think I have one, but then that's a personal feeling. And there's also no leadership in the area. So, in fact, what we have here is a major disease, which probably is infecting about 1.5 million people per year, which has no strategy and no leadership. So, of course, nothing's getting done. So, actually, that is the part, and I haven't got any other slides on this, you'd be glad to know, that this is the first thing that needs to be done. Secondly, if I go around the world, and I've counted them, people who have labs that are set up to do cutaneous leishmaniasis, there are 3 in South America, 2 in Europe, and 1 in Iran. And they're the only groups who I really think have any possibility of making... Obviously, there are lots of labs who work on cutaneous leishmaniasis, immunology, biochemistry, etc., but actually have any significant effort at the moment to actually develop a new drug for cutaneous leishmaniasis. I didn't mention the other one, which is the 7th one, which is the Walter Reed Army Institute for Research, and I'll come on to why I don't mention them in a minute. But I want to take you through some of the issues which I think, and the opportunities again, which relate to this. So it's only 7 labs around the world who actually, I think, actually have the skill sets and the ambition to get a new drug out for this disease. And one of the things is about this strategy, we already have to decide whether we want an oral or topical, how many species we're going for. And my view of the strategy is if we only say, well, we've got a wonderful drug here, it only works against Leishmania, major infections, and I go to Sanofi, or Merck Serono, or Janssen, they'll say, well, forget it. You know, what are you going to do about the other 14? It's a ridiculous concept. And that's been the approach so far. Also, we have to also think about the patient-friendly formulations and how to treat patients like this. This is not a disease which is uniform, again, in terms of its presentation, how to treat them in primary healthcare centres, and also to treat, or rather to change the behaviour of people, because that was mentioned in the panel before. Actually, patient treatment behaviour change is important. So if people know that there's They don't have to be injected into a lesion with antimony. How will that change their behavior? If they think they have a cream, we don't really know how that will change their behavior. But one of the great proposed new treatments is a topical treatment for cutaneous leishmaniasis, which is paromomycin, which is this aminoglycoside that's been known since 1984 to have a topical route of efficacy. And the latest clinical trials published in the New England Journal of Medicine in 2013, I think, illustrate three of the major concerns I have. I don't know, I won't criticize anybody who refereed this paper, but I don't know how this paper got into this journal. Because A, paromomycin is really only effective against Leishmania major infections, doesn't have really much activity against any of the South American infections. But if you read the paper, it looks quite good. But if you go into the supplementary materials, you find out how this treatment is delivered. Firstly, take your patient, take the lesion, take the crust off the lesion. Are you going to do that on the nose or the fingers? No. Take your crust off the lesion, wash out what's underneath, apply your treatment, your paromomycin ointment, and then put an occlusion on the top. Now, you can see this going down a bomb in a village in Afghanistan or Guatemala, can't you? So the other thing is they call it a topical formulation. It's not, because actually when you take the crust off, it exposes underneath the bed of capillaries. So in fact, you're putting a treatment on, it's going straight into the circulation. It's a transdermal, it's not a topical. It's not actually getting across the skin and killing the parasites in the area where they need to be killed, which is in the dermis underneath the lesion. Actually, I've been banging on about this for years, so sorry for those of you that have heard me bang on about it before, but it is a critical issue of misleading the direction and the strategy for the development of a new drug. And I've been fortunate in having two PhD students who've both worked in the cosmetic part of the industry before they've come to work with me. And the cosmetic industry know how to get anything across the skin, you know, titanium to protect you from sunburn, anything, they can do it because they pay a lot of money to do it. And we've been looking, just going back to the basics. We take nodules, so before they ulcerate, we take infection and we take the skin of mice from the uninfected area and from an area where there's a nodule, hasn't ulcerated. And actually all we've just done is this basic skin pharmacokinetics. The skin over the lesion, or what is not a lesion, it's still an early infection, is far more permeable to the classic ibuprofen and also testosterone and all these classic skin measures, parameters when you measure skin permeability than uninfected skin. And it's also, apart from amphotericin B, which is the most impossible drug to get across skin at all, apart from amphotericin B, it's actually all the other anti-leishmanial drugs also get across the skin over an infected site due to the edema, due to the change in the nature of the inflammatory response. It encourages drugs. So why don't we just take this sort of approach and actually do proper topical formulations rather than crust removal and all the rest of it? So I have a real problem about the direction at the moment of some of the cutaneous leishmaniasis. The other subject which is always on the tips of people's tongues at the moment for curing cutaneous leishmaniasis is not just killing parasites, but how to accelerate self-cure. And there have been a number of immunomodulators proposed over the years and also therapeutic vaccines. But I've just taken some quotes from clinical immunology papers which actually have been published in the last few years. Cutaneous leishmaniasis is not one form. Whether you've got Leishmania brasiliensis, Leishmania tropica, Leishmania major, and actually you do the immunocytochemistry, you do the cytokine profiles, they all vary, and there's mixed responses. Most of them have mixed Th1, Th2 responses. So you use your model mice, which is a Th1 or a Th2, and you can do anything you want with an immunomodulator. But actually patients aren't like that. They're complex individuals. There are complex immune responses that depend upon individuals and depend upon the species of parasite that's causing that infection. So I started off thinking that immunomodulators 30 years ago were the great way we'd move forward. But now, not only do I think there's no panacea, but actually I'm incredibly cautious about people who propose immunomodulators because, again, what form of disease, what parasite are you going to be using against? It could cause antagonism, it could cause agonism, it could accentuate the infection. And just looking at one TLR7 agonist, which is on the market, which has been tried, imiquimod, in conjunction with antimonials, a clinical trial against Leishmania entropica in Iran, no interaction at all, but against Leishmania brasiliensis improved. Yes, there was some adjunct activity of the immunomodulator plus the antimonial drug. So I think there's a big warning here that we ought to take this sort of approach, deal with it with great caution. And I'm actually far more now in favour of what I call the small molecule approach to this disease in the right formulation, whether oral or topical, than taking this approach. So I come up to the last slide, and I think apart from this clear strategy, leadership, and all these issues which I think are sadly deficient for this disease, I can actually point to one thing that Piero Oliaro and colleagues at TDR have actually set about a job which needed to be done, which was defining properly how to do a clinical trial for cutaneous leishmaniasis, which had never been done before, and how to actually define set endpoints and sort of how do you monitor with biomarkers and diagnostics. So that has been done. And I just wish for Piero and TDR's point of view, they actually had something to test. So it's wonderful to have great endpoints and designing clinical trials. Actually, what we're missing at the moment, and that's my appeal to Robert and to others working in the field, to actually give them something to work on in clinical trials, because at the moment there is nothing. So thank you for your attention. Thank you.